to actually stop the leakage of blood out of the damaged vessel. Obviously, in larger vessels, especially larger arteries that have the pressure of the heart behind it, that's not going to be sufficient. Okay? So the next step is platelet plug formation or platelet aggregation. And we haven't talked about platelets yet as our formed cells, but we'll look at them when we move to step 2. And chemicals released by platelets can enhance the third step, which is the formation of a blood clot. And our word for a blood clot formation is coagulation. So as a clarification, we used a term yesterday, agglutination. I don't want you to confuse them, okay? Coagulation and agglutination are two entirely different events. So agglutination is the process by which antibodies clump 2 or more molecules together. So that's what was happening with our red blood cells. It doesn't have to be just with red blood cells. If we have a cluster of structures, each of them bound to the 2 aspects of the arms of the antibodies, and they're clumped together, that's agglutination. And another word for our antibody, remember, was an agglutinin. Coagulation is the formation of fibrin threads. So the end event of coagulation is protein fibrinogen being converted to fibrin. Both platelet plug formation and coagulation are a series of cascades, are enzymatic reactions. So a cascade is the opposite event of negative feedback. Negative feedback, when something happens, we have an increase, that results to slow down whatever triggered that increase. With a cascade, we have an event that triggers more of that same event and more of additional events to get to an end result. All right, we're going to be looking at 2 types, 2 components, well, 2 events that are identified as positive feedback. In this unit, homeostasis is one, specifically coagulation. And then the next event is the formation of complement, part of our defense system that we'll be talking about next week. So the first event, again, requires a cutting of the vessel. You don't usually see vascular spasm with, say, atherosclerosis or an irritation of the lining of the blood vessel. So we have that first event. And like I said, in small arteries or arterioles, that may be sufficient to stop blood flow. The second event is the platelet aggregation or platelet plug formation. And so before we look at that further, I want to talk about platelets. So here's our damaged vessel. This would be the tunica externa, tunica media cut in cross-section of the smooth muscle cells, so we have a long view of our vessel. The endothelial lining of the tunica media. A cut has occurred, and these purple cells are the platelets that have started to stick to each other and fill in that gap. So what are platelets and where do we get them? They're a part of our formed cells. Okay, we have several 100,000 of them rather than the millions of red blood cells that we have, or the 5 to 10,000, per cubic mil that we had for the white blood cells. But they are not a whole cell. They never were. So our erythrocyte was a whole cell that lost its nucleus and many of its organelles. That it was just basically a bag of hemoglobin and other enzymes and additional enzymes. Platelets are derived from a large cell, about 2 or 3 times, well, 3 to 2.5 to 3 times the size of a monocyte. So 60 microns. Remember, our monocytes were about 20. So diameter-wise, volume-wise would be even more. It may have one or more nuclei. And the plasma membrane is enfolded almost like a corn maze over and over and over and over and over. So we have multiple layers of plasma membrane. And regulated by a hormone made by the liver, thrombopoietin. Remember the erythropoietin that the kidney made that increased red blood cell formation? That increases both the number of megakaryocytes and the rate at which they release platelets. And this plasma membrane will pitch off into small sacs, and those are the platelets. So if a red blood cell is this size, platelets are that size.
Can you please repeat that function of the
Thrombopoietin? So it's going to trigger additional megakaryocytes, so mitosis of the stem cells of megakaryocytes, and then it's going to increase the formation of these vesicles. So we're going to have more mother cells and more babies.
And the weight and the rate at which they are released is that?
Yeah, so the platelets are formed by the pinching off of the membrane of the megakaryocyte, so that pinching off rate increases. So we have more sources, more megakaryocytes, plus we have more platelets being formed, a higher rate at which those are formed.
They're fragments of cells.
Yeah. So the megakaryocytes are too large to actually leave the bone marrow. The platelets easily leave the bone marrow. They function in the blood, so they're not going to be exiting the blood vessels, the capillaries, and going into the connective tissue. So their role for hemostasis is within the blood itself. Remember how our white blood cells did diapedesis? And our red blood cells and our platelets stay in the blood, unless, of course, the vessel is cut and they pour out. And they last in the blood for about 10 days. Okay. Obviously, they're not going to reproduce. They have no structure to do so. These sacs of platelets contain several different molecules. So they're listed on this slide, and I'll write them up here on the board. So within the platelets, we have serotonin, ABP, and thromboxane. Remember that's made from epoxidants. Thromboxanes are made from arachidonic acid. And also there are fibers, myofilaments of actin and myosin, because the platelets kind of shrink among themselves. So the platelet itself kind of contracts when it becomes activated. So serotonin, ADP, and thromboxane, and actin and myosin are some of the major components of the platelets. I don't know if you have this illustration
Which
in your lecture
PowerPoint?
notes.
Which PowerPoints are you looking at?
Which one?
It's under hemostasis and blood typing. Be at the beginning of the blood typing ones. So here is the cut vessel wall. All right, it doesn't necessarily have to be cut, but the collagen fibers can be exposed due to irritation and inflammation. And the platelets come in contact with those collagen fibers. There's a molecule here, the von Willebrand factor, that interacts between the platelets and will bind to them and cause them to become sticky. And when they become sticky, they bind to other platelets and they release these chemicals. So serotonin, the same molecule that we know as the neurotransmitter, the elevated mood neurotransmitter, is also a vasoconstrictor. So when it's released here, it's going to cause these smooth muscles to constrict and enhance that vascular spasm that we started out with. ADP increases the platelet stickiness, so then they release more of these chemicals. And thromboxane does both. Should be thromboxane A2. Does both. All A little bit farther into the lecture, when we talk about how we prevent platelets from forming, you probably heard that aspirin functions to thin the blood. With my husband's family history of coronary problems, he takes a baby aspirin, 82 milligrams every morning. And so the role is, well, it thins the blood. And based on our discussions, you might have the idea that it adds more fluid to the blood, makes the viscosity less. It doesn't do any of that. It keeps the platelets from sticking to each other by inhibiting the activation of thromboxane 2, A2 and its synthesis. So aspirin works on this aspect of platelets. So not even the coagulation steps. But, so clots can form from platelet activation either by sticking to the collagen, all right, or we'll talk about deep vein thrombosis. So platelets settling out because the blood isn't moving cleanly through the veins, or in the heart, we talked about atrial fib. And in the atria, the heart contraction is not significant, just a quivering. And so in the pectinate muscles of the atria, the platelets can settle out, and as they come in contact with each other, start to get sticky and trigger clot formation as well. Remember, clot formation is the filament component that is added to this. So up here we don't see those filaments yet. All right. Now, over here on the side, I added to this slide, it's not added it yet this morning, it's not in your lecture notes. I added two molecules, prostacyclins and nitric acid. And those are made by normal endothelial cells that haven't been damaged. Because we don't want clots forming just anytime, anywhere. And so those are released by normal blood vessel cells and help to inhibit the platelets from adhering to the vessel wall. So you notice that's to the side of our damaged vessel wall. And that helps to prevent the platelet aggregation.
Inhibit the platelets to adhere to the
Vessel wall.
vessel wall.
That's nitric acid and prostacyclin.
Thank you.
So as the platelets become sticky, remember I said they had actin and myosin? By becoming sticky, I'm not just talking about a glue. They send out those receptors that connect to each other. Okay, von Willebrand factor is part of that connection. They constrict, they contract. I don't know if you remember epithelial tissue in the stratum spinosum. And the fact it was called stratum spinosum because in the preparation of the slide, the cells shrink and the cell junctions make them look spiny.
It does look nice.
So when actin and myosin contract or constrict because of the actin and myosin, that exposes more of fibrinogen receptors, and fibrinogen can bind to the platelets. And fibrinogen is the precursor to a fibrin filament for coagulation. So there's a lot of overlap between platelets and actual coagulation. They don't require each other, but they each enhance the other's activity and presence. So here's another image that I added this morning. On that previous slide, on that previous slide, you can see the von Willebrand factor right here. So it can be released from the connective tissue. It can also be released from the platelets. All right, and so this next slide just shows their activity here in this separated component. And then they'll interact with the platelets and help to enhance the platelet activity at that injured site. This is a new slide as well that just shows the same thing as the two previous slides and has all those molecules in place. So at the top, the VWF is the von Willebrand factor, and you can see it connecting between the platelets, and then additionally here as well. And these are the other molecules that are released by those platelets when that happens. So now we have our platelets attached to each other, a growing clump of platelets, and again, that might be enough to block small blood vessel breaks in their integrity. But the structure of the platelet plug is enhanced by the formation of these lacing filaments, the fibrin threads. So last semester I talked about my favorite biological material, which is Jell-O. And we used it to illustrate cartilage entrapment and how glycosaminoglycans and glycoproteins can trap fluid and create a semi-solid gel of the cartilage. And that essentially is what's happening in our next step with coagulation. So coagulation adds to our platelet plug these long threads. So in Jell-O formation, the sugar of course enhances the taste, but when you open a package of Jell-O or some other brand of gelatin, and you add hot water to it, it's very fluid and flows easily. But as it cools, what happens to the viscosity of the fluid? It's very thick, it stops flowing. All right, and it's not because of the evaporation of the water content is still there. Well, it's not the temperature either that enhances the rate at which this happens. But what happens is, because you could add cold water to Jell-O, and it doesn't
add ice.
Well, but if you just add cold water to the gelatin, you're not gonna have Jell-O form. You have to add the hot water first. And then as it cools, you see this change. It's increased by the cooling. So what happens is the heat causes the Jell-O protein, the gelatin, to polymerize into long strands instead of a powder form. And those long strands are interlaced like a felt. The water molecule is trapped within those. And if it can't move, our liquid phase of the water changes to a semi-gel. So, yes.
Can you see that in a microscope in Jell-O?
Strands as before you add.
I don't know if the resolution is soluble within that fluid, so I don't know if you could see it like we were looking at yesterday with the coagulation component or not. So that can occur without the platelets. Remember, we can spin the cells down, and we're left with a fluid known as plasma that still has the clotting proteins and can still coagulate. So you may have had an injury where it didn't bleed, but you could see the tissue fluid kind of ooze to the surface. You didn't have cells, but it thickened and kind of formed a jelly-like material, even though there weren't cells in there. So plasma can coagulate, because we have to take the clotting proteins out to form the non-clotting serum. So red blood cells and platelets are not required for that coagulation phase to occur. They can enhance it, and the fibers being present strengthen the platelet plug, so the whole thing works better together to block blood leaving the vessels, but they are not required. So you have this diagram in your lecture notes. It's kind of scary. I already told you, reading a chart like this where I don't know what factor 3 and factor 7 and factor 12 and all of that stuff, it's hard for me to read names that don't mean anything. But these were, and to compound the problem, both with this and with complement activation, these are numbered, and complement is given letters, not in the sequence of order in which they're activated, but in the sequence in which they were identified. So they're all completely, the numerical order is not significant to the order of function. So I'll show you what I want you to know. I'll go through the steps, but you don't have to know every identical step. All right, and I'll tell you what I want you to know. First of all, in this diagram, you can see that there is an extrinsic pathway and an intrinsic pathway. What we're talking about is what the origin is of the trigger in the activation of the clotting factors. The extrinsic pathway requires damage surrounding tissue. So you cut yourself. You had to cut the epithelium and the connective tissue before you got to the vessel wall. Or crushing bruise. So you go banging against the corner of the door or the doorknob or whatever. A few years ago, we used to have on our office doors and all the classroom doors, door locks, stops that you could push down. So if we were carrying a tray of slides or whatever, models from one room to the next, push the door on, bringing the cadaver tray out, push the door, unlock it, and carry it through. Well, on one of our campuses, there was a lawsuit brought by a disabled individual in a wheelchair, and I don't know if the sidewalk was narrowed because the door was propped open. I mean, my office is a lot easier to get into. You open the door and then you have this wide doorway. But anyway, brought a lawsuit, and so they went to all of the, maybe it was that lawyer that goes to all the places and files lawsuits on apparently someone else's behalf, but he gets the money. They went through all of the doorways and removed those door stops. And so the little door stop that I use to prop the door open to bring the cadaver in is actually not supposed to be there. So we hide it around the corner and just, so shortly after this happened, I was bringing in a tray of something into our classroom and I couldn't hold the door open. So I pushed it open and then tried to walk through really fast before it crashed on me, and the door hit me right on the side of the hip and made this big bruise. So I went to show the secretary and I went, look at this, what happened to me because of this stupid rule. And she said, let me take a picture, we can send a notice to whatever. But, so that's a bruised tissue, all right? The blood vessels were damaged and the blood seeping out into the connective tissue, so the clotting is occurring in the tissue as well as at the vessel wall. That's the extrinsic pathway. It requires the addition, or occurs because of, the addition of a chemical from those damaged cells outside the lining of the blood vessel wall.
So any kind of damage you're saying, and not just a cut, it can be
Right.
inside your throat.
Okay? That requires the addition of this tissue factor, this tissue thromboplastin. That's the key event of the extrinsic pathway. It's also a shorter pathway, fewer steps. The intrinsic pathway may have damage to the lining of the blood vessel wall, but doesn't require additional chemicals from cells outside the blood vessel. So there might be exposure to collagen from the tunica intima. It might be an inflammation, so phlebitis is an inflammation of the veins. This occurs if you take blood from a phlebotomy and put it in a glass tube. The glass tube surface is foreign, it has different charges than the blood vessel wall. That will trigger, the key event is that all of these trigger the activation of molecules already within the blood. So we're not adding anything to the blood like we are with the tissue thromboplastin. The key difference in the actuality of the start of these two pathways. There are more events that occur within the blood, primarily because we don't want this happening just by chance. Obviously, if you have a damaged vessel, you need to have a clot. But we don't want the blood just flowing around a corner and saying, I didn't like that corner, I'm going to start clotting. Okay, so we have additional steps that have to occur. They all eventually reach a common activation of a common factor. So from that point on, this is identified as the common pathway.
From stage 2 on?
Pardon? The factor 10. So I, this morning, took this pathway and made some additional slides, just took 3 slides, took these some information and put it, made the factors colorful, but broke it down so it wasn't quite so overwhelming. So this is looking at the intrinsic factor. It's based on the same slide that you already have, but looking just at the intrinsic factor. So what I do want you to know from this slide is the very first factor that's activated is factor 12, also known as Hageman factor. And what activates it are one of those 2 changes. Both of them are a different surface. Either the surface of the vessel wall that's been damaged or a new container. They have that in common. There's something different about the surface of the vessel wall. And that triggers the factor 12 to become an active factor 12. So I want you to know that that first molecule is factor 12. Okay? Or if I ask you the significance of factor 12, that it is the one that's activated by contact with a damaged surface. That then changes inactive factor 11 to active. And along with calcium, factor 11 changes inactive 9 to active factor 9. Those numbers in there, you don't have to worry about the order. Okay? We'll deal with factor 9 in just a moment. Factor 9 is going to activate factor 10. But I do want you to recognize, so on this page, factor 12 and factor 9 will be important. And I want you to recognize the significance of calcium. You don't have to know where, between which steps calcium is required, but I want you to know that many of these are enzymes, and calcium acts as like a coenzyme in the activation of some of these clotting factors. So without calcium, clotting, coagulation is inhibited. So molecules that remove calcium from the blood are known as chelators or from any solution. So a chelator binds to and removes calcium ions from solution. So anything that removes calcium is going to cause the blood to take longer to clot, or even prevent it from clotting. Okay? Now I told you yesterday that I have a student in my first section who has polycythemia. They don't know the cause. It's not dehydration or blood doping. It's a disease. And she goes in every 5 to 6 weeks to get her blood drawn, kind of the reverse of blood doping. They take her cells out, take the blood out, spin it down, remove the blood cells, add the plasma back, and throw away the blood cells. Or maybe give them to somebody else that needs blood cells. But her blood is too thick because of the polycythemia. And because of these extra cells that the liver has to break down, she has too much iron being released from the red blood cells, hemoglobin. And so she develops, when it gets close to the time when she needs another bleed, she'll start to develop a slight case of iron poisoning, and she said her skin starts to itch. And so she found that an effective removal of the iron are the same molecules that remove calcium, and she can take by this over the counter. It's called EDTA. It can be produced from plants. And so she started taking these chelator pills. And when she started getting the itching, and the first time after she took them, she went to get her blood drawn, and she said her blood just poured into the vacuum tube. And when they took that out, it kept pouring out of her arm.
Oh my goodness.
And it wouldn't clot. And so the phlebotomist was like, what have you been doing? And so she said, well, I'm taking this new pill that helps to relieve the itching. So she realized very quickly. So now she will, if her skin starts to itch, she'll take that medication but stop it before she goes in for the bleed. Hopefully she stops it also if she has to have surgery or anything like that. So it's not removing her clotting factors, it's not decreasing her platelets, it's taking the calcium levels down in her blood and thus inhibiting the factors from being activated. So we get, within intrinsic pathway then, we get to factor 9. All right, so kind of hold that in your mind. You can see it on the diagram in your lecture note. Because factor 9 is going to be the intrinsic pathway's method of activating the beginning of the common pathway, which is factor 10. If we go back to this slide, we've come down this pathway here. Here's factor 9, and it's going to activate factor 10. Now we're going to look at the extrinsic pathway, which is much shorter. All right, doesn't have as many steps that need to be regulated. And so this is extrinsic pathway. It requires tissue damage and the release of tissue thromboplastin, also known as factor 3. I hate vague terms like factor 3 and 4 and 5 and 7, so I'm going to call it tissue thromboplastin. You do not have, I'm not going to test you and ask you what's another name for factor 3. All right, so I just am going to use tissue thromboplastin. That is released by the damaged cells. And along with a factor in the blood that if the blood vessel is damaged, seeps out into the connective tissue, forms a complex. Again, calcium is needed for that. So this complex, along with factor 9, will activate factor 10. The complex will always have factor 9 because that's in the blood. All right, so those two will always work together. Factor 9 does not need this complex because the blood will coagulate in the glass tube and there is no tissue thromboplastin in that glass tube. So factor 9 can still be created like we saw previously, and factor X can be activated by factor 9 without any of this occurring. But when we do have tissue damage, then this complex and factor 9 together will activate factor 10. Is that clear? So the extrinsic pathway will use factor 9 to activate factor 10, but the intrinsic pathway doesn't need to. Okay. Here's a place where platelets also contribute and enhance the function of factor 9. These are lipids, phospholipids from platelets that, along with calcium, are used in the activation of factor 10. Again, they don't have to be there, all right, because plasma will coagulate without the platelets being present, but they enhance, decrease the time in which that occurs.
PS stands for what?
Platelet factor. So going back to this diagram,
What's TF then?
pardon,
TF,
tissue factor,
tissue factor.
thromboplastin.
Can we go back to the others?
Let me just bring us together through here. So this is our beginning of our common pathway. So the tissue factor, thromboplastin, tissue factor the same, forms a complex with factor 7, and in the presence of calcium, will activate factor, and the presence of factor 9 will activate factor 10. Without the tissue factor, we need to go through a series of other events to get to factor 9 to factor 10. But this then is our common pathway, and I'm going to write that on the board. You want to go back to what slide?
The one before.
The extrinsic.
The previous,
This one or
the
the?
extrinsic. Thank you.
These are not in use right now.
No, I made them just a minute. I finished just 30 seconds before class start, but I can put the revised ones up as that.
They stocked 1,000 more sheets of paper on the printer, so.
Okay?
1 second.
Okay, so the little arrows pointing in between the active 9, it just means.
These are just
enhances-
additional components.
It's a
Okay,
9 to get.
not required, but it enhances the property.
What's PF again?
Calcium required.
Calcium's required. Okay.
So that calcium goes out.
Okay?
Thank you.
Okay, this is information is also on the diagram in your lecture notes. It's just, I broke it down and enhanced it with color. So now we have the common pathway. So we have factor 10. Again, this is in the blood. And as we'll see in a moment, many of these factors are made by the liver. They're proteins, they're enzymes, and it's part of the synthesis function of the liver. So calcium, again, is required. Our platelet factors and factor 5 enhances this. But this is going to activate an enzyme known as prothrombinase. Which is in its inactive form in the blood. And this enzyme converts prothrombin to thrombin. it's that molecule that has a few extra amino acids attached to the end of it. And so what the enzyme does is increases the separation, the removal of those extra enzymes. And then we have the active molecule without it. And so that's thrombin. And then thrombin functions as an enzyme to convert fibrinogen, cause the polymerization of fibrinogen, which is more of a globular protein, to long filaments of fiber. And that's our end result.
So is that like what a scab is?
The dried, yeah. It doesn't have to be dried for the fibrin to be there, but as the rest of the fluid evaporates, that's what you have there. Now, when we were doing body fluids in lab, we would test clotting times. And clotting times are the time it takes for blood to clot. There's other ways of measuring it. It says the same thing on the slide. There's prothrombin time, there's partial prothrombin time, and those will measure different components of this. But in the lab, we take that same capillary tube, draw blood up into it to about the full length, wait. Most most clotting times are 2.5 to 3 minutes. So wait a minute and a half and break off the end. It would break cleanly, and the blood would still be wet. And then you'd wait another 30 seconds to 2 minutes, break it again. And if you had a short clotting time, you'd start to see a few strands connecting the broken ends of the glass. And by 2.5 to 3 minutes, when you broke the glass, they would be like a little plastic, it's kind of stretchy, connecting the two ends, and a clot had formed. And so that was again another test that students could do with their own blood to do a dirty.
So those two fluids have heparin.
No. If they did, obviously,
It wouldn't-
that wouldn't happen, yeah. But good question. All right, so some of these molecules I talked about are also identified as factors. As I said, I have a hard time remembering, is it 3 or 7 or 5 or 6? So fibrinogen itself is called clotting factor 1, meaning it was discovered before the others. But we're just going to use those terms rather than the numbers. Notice how many of these are synthesized by the liver. Look at the role of vitamin K. You know, anybody remember where we get our vitamin K? It's a fat-soluble vitamin, so it's been enhanced in milk, but no, you're close. It is part of the digestive factor. Intrinsic factor was the absorption of vitamin B12. Vitamin K is due to the symbiotic relationship we have with E. coli in our large intestine, in the colon. We don't have enzymes that break down cellulose, so that isn't absorbed by our body, but the bacteria do. And they synthesize vitamin K. So, along with water in our large intestine, we absorb vitamin K. And it's one of the, when I worked in the newborn nursery, a baby would come to us from labor and delivery, and we'd give it a bath. We'd put drops of silver nitrite in its eyes in case it had been exposed to venereal disease, and we'd give it a shot of aquamaphytin, which is vitamin K, in its anterior thigh. They don't have enough muscle tissue in their arms or their buttocks, so we go in their tiny short little needle into their anterior thigh, and aquamaphytin is vitamin K. Because as newborns, they don't have any E. coli in their digestive tract yet. And so that takes a few days to build up. So the liver makes prothrombin, but it uses vitamin K as a coenzyme to do so. And prothrombin was the molecule that is activated in those steps over there to make thrombin. So calcium even gets its own factor name. All right. So this is the Hageman factor. Calcium ions, tissue, thromboplastin, all those have their own factor numbers, but we're just going to use the names for them. The main reason I put that slide up there was to recognize the liver's role. It makes it a bit of a contradiction, a conundrum. Liver transplants are done because people are in liver failure. It's a surgery, And yet, they're missing a bunch of clotting factors because their liver is in failure. And so it's a bit tricky. They have to be treated, they have to be given these clotting factors prior to the surgery. Usually in a plasma, kind of like a hemophiliac, would go in for, they don't need all the red blood cells, but they need the clotting factors. So again, a transfusion of the plasma. So some people are plasma donors, they'll give blood, they'll spin it down, take the plasma, and then they'll add the red blood cells back. But you have to be careful, that's going to increase viscosity as well, but their body will quickly adjust the blood volume. So when they're taking plasma, they're often using these clotting factors, is what they're taking